Hello everyone. In the last few videos, we spent time understanding the male and female reproductive walls of the flower, that is the androecium and the gynoecium. In this video, we will try to understand the interaction between both the pollen and the pistil, the double fertilization which occurs after that, development of endosperm and development of embryo. So last time we learned that a female unit known as a carpel consists of a stigma, which is receptive in nature, a style and an ovary. So when a pollen grain, which is a microspore formed in the microsporangium, the androecium, comes and lands on the receptive stigma, it can have two outcomes. If it is a correct pollen grain, that is, it is a pollen grain from the same species of plants, it will germinate, and if it is not, the pistil, the carpel, will reject it. So in this case, let's assume it is correct pollen grain. So once the pistil recognizes that the pollen grain is the correct one, it will send chemical cues for it to germinate, and the pollen grain forms a pollen tube through which the male gametes pave their path to the ovary. Now, in this case, the pollen grain lands on the sticky stigma. If wrong type lands, it is rejected by the pistil by preventing germination. Growth of the correct pollen grain is guided by chemical cues which are sent by the synergids, which are present on the micropylar end. The interaction is therefore dynamic because there is a lot of action going on here. So all the events from the deposition of the pollen grain to the entry of the pollen tube in the ovule, that is from pollination to fertilization, is known as a pollen-pistil interaction. So here in this image, you can see there are many failed pollen grains which have failed to germinate. The pistil did not allow them to germinate, and this pollen grain won because it produced a pollen tube and it has successfully reached the egg apparatus. This came along the way in response to the chemical cues sent by the synergids. Okay, now let's talk about what happens or how does the pollen tube enter into the ovule. So we have a pollen tube which can enter either from the chalazial region, from the micropylar region, or from the integuments. Now, if a pollen grain germinates and the pollen tube enters through the micropyle, it is known as porogamy. If the pollen tube pierces the chalaza and enters through the micropyle, it is known as chalazogamy. And if the pollen tube penetrates the integuments and then enters through the micropyle into the ovule, it is known as mesogamy. So pollen grain germination can also be induced in vitro artificially too, which means you can take a pollen grain and without a stigma, you can trigger it to generate a pollen tube. How can this be done? You can use sucrose and boric acid. Sucrose is used for giving nutrition and boric acid is used for enhancing. Artificial hybridization is one of the preferred methods and is accomplished by bagging and emasculation. Artificial hybridization is when you have a pollen grain which is selected from a selected plant and you want that pollen grain to fertilize the pistil. So you choose the pollen grain and you artificially place it here. Emasculation is the procedure where you cut off the androecium before they mature so that the pollen grain cannot travel via vectors by pollination. And bagging is after the female gametophyte has matured, you put a plastic bag around it which will prevent a pollen grain from coming and attaching to the stigma. Now, did you all know that angiosperms are the only division of plants which display double fertilization? Now, let us learn about double fertilization. So, when we saw that the pollen grain correctly matched with the pistil, and by means of cues generated by the synergids, it successfully reached the ovule. Once it penetrates the ovule, the synergids have a lot of water. The pollen tube absorbs the water and it bursts. When it bursts, the two male gametes that it has, one of them goes to the secondary nucleus and the other one goes to the oosphere, which is your egg cell. So, initially the secondary nucleus had 2 nuclei. Now it has 3 nuclei, and hence it has become a triploid entity, and the oosphere is now a diploid entity, one maternal and one paternal nucleus. So the tip of pollen tube absorbs water from the synergids and it bursts open. This releases 2 male gametes. One haploid male gamete fuses with the oosphere and other fuses with the secondary nucleus, as we discussed. So there is triple fusion going on in the secondary nucleus, and hence it forms the endosperm in future, and the oosphere, which has been fertilized by the male gamete, forms the embryo. So there are certain features of double fertilization that we need to know. When the male gametes are introduced by the pollen tube, this phenomenon, this process is called as siphonogamy. Siphon means a tube or a pipe. So when the male gametes were brought by a pollen tube, we call it siphonogamy. Then the parent plants invest seeds with food only if the egg is fertilized. So if there is no egg apparatus present, like the oosphere and the synergids will not release chemical cues. The synergids will not provide the pollen tube with water, which means there will be no fertilization. And hence, the endosperm also doesn't form, because in the process of fertilization, we have another male gamete which will come and fuse with the secondary nucleus. So the plants invest seeds with food only if the egg is fertilized. It restores the diploidy, because oosphere or your female gametophyte was only N. You need a 2N to multiply and form a new plant. So here we have one male nucleus and a female nucleus. They fuse and they give rise to 2N. Both the male gametes formed are used up, hence less chances of polyembryony. Now, just in case we just had one male gamete which fertilized the oosphere, it would have been an open window for the other male gamete to fuse with some other cell and form an embryo. So that is called as polyembryony, and it can have risky conclusions in plants. Hence, both the male gametes are used up here, one for production of endosperm, another for the production of embryo. Now that we completed with double fertilization, it led us to the result of a primary endosperm nucleus, which is triploid in nature, and a fertilized oosphere, which is diploid in nature. Now, the primary endosperm nucleus develops into an endosperm. What is endosperm? It is a nutritive tissue which forms by repeated divisions of the primary endosperm nucleus. So each of these nuclei are triploid in nature and they totally surround the embryo because the embryo needs to derive nutrition from them. It provides nutrition in the form of starch and other nutrients, one of the most simplest forms of food. And then there are three basic types of endosperms based on their mode of formation, which we will be learning further. So initially we had a secondary nucleus which is diploid. It was formed in a mature female gametophyte. Then post double fertilization, it gave rise to a primary endosperm nucleus which was triploid in nature, and further on finally it gives rise to an endosperm which is the nutritive tissue used by the embryo to mature. Now the first type of endosperm we see is nuclear endosperm. Nuclear endosperm, the primary endosperm nucleus undergoes cytokinesis, which is division of the cytoplasm. Nuclei are pushed towards the periphery, which means there is no karyokinesis until the nuclei are pushed towards the periphery. So all the nuclei you see here do not have any cell walls around them and they have been pushed to the periphery, and a vacuole is formed in the center. When the nuclei are pushed, that is the time they start karyokinesis, which is development of the cell wall, and finally the endosperm can be called multicellular because there are many cells present here. Sometimes what happens is this karyokinesis, it remains incomplete, so only in some peripheral area it will have cell wall valley cells, and in the central region it can have without cell wall cells, so only nuclei floating around. This is a typical case in coconut. So in coconut we have white flesh which is solid endosperm. This is the karyokinest cells, they have formed the cell wall, and in the central region we have liquid endosperm. This is formed due to the deposition in the vacuole, and these cells did not form any cell walls. And it is seen in about 161 angiospermic families. The second type is the cellular endosperm. In this, if you see, each division ends in karyokinesis. It does not stop at cytokinesis. So as soon as the cell starts dividing, it forms various cells, and hence it becomes a multicellular structure in no time. So from the beginning, it is a multicellular structure. It is observed in 72 families of dicots, and these are petunias. So in this, it is not really as conspicuously visible because since coconut has a huge fruit, we could observe it, but in petunias you don't have huge fruits, so it is tough to observe it. Then we have Hilobial endosperm type. In Hilobial, what happens is the first division is transverse, okay, and it ends by formation of two unequal cells. So this is the chaliceal end, this is the micropylar end on the side where the embryo is formed. So on the micropylar end you have the larger cell, whereas on the chaliceal end you have the smaller cell. The cell on the micropylar end will start dividing and redividing and will start forming cell walls. So it will become multicellular on one side and it will remain just in an akaryokinase manner on the other side. So it is a mixture of both nuclear and cellular kind of endosperms. This is normally observed in Asphodylus Similarly, here it is tough to observe it because it doesn't have really huge fruits, and because it is observed in all a Hilobial order of monocots, it is known as Hilobial endosperm. We're not done yet. We still have to form the embryo. Let's see how is the embryo developed. So after the male gamete has fused with the oospore cell, it becomes a diploid cell, and hence this oospore is now diploid in nature. This had two laterally arranged synergids initially, but now we're going to just talk about this oospore for the further development of the embryo. So the oospore divides once transversely to give rise to two cells. So one is the larger cell and the other one is a smaller cell. The smaller cell is towards the callosal end and the larger cell is towards the micropylar end. The smaller cell is known as the embryonal initial cell, whereas the larger cell is known as the suspensor initial cell. It is also known as the basal cell because it is present towards the base, and the embryonal initial cell can also be known as the terminal cell because it is present towards the callosal terminal. Okay, so now we talk about the further divisions that occur in these cells. So the callosal end is still hosting the embryonal initial, whereas the micropylar end is hosting the suspensor initial cell. Now the suspensor initial cell divides to give rise to around 6 to 10 cells, and these cells are filamentous in nature. Now the cell towards the micropylar end really grows in size and it is known as the hostorium. It is really huge and bulbous, and this is the cell which presses into the endosperm inside the ovule. Then the cell towards the callosal end or the terminal end is known as the hypophysis. Now that we are done with learning about the suspensor divisions, let us talk about the divisions which occur in the micropylar region. A few divisions have occurred here, but let's talk about a little further division structure. So the micropylar end, as you can see, the suspensor cells have grown and given rise to a hostorium and a hypophysis, and this is the embryonal initial cell. It has reached the heart-shaped stage here. So as the suspensor initial cell undergoes division, similarly the embryonal initial cell also undergoes divisions. So it specifically undergoes 3 divisions. So in the first division, it gives rise to 2 cells. In the second division, it gives rise to 4 cells. And in the third division, finally, each cell will give rise to 2 more cells, which means finally it will form 8 cells. So 8 cells form an octet. Now we have 8 cells which are arranged in an octane structure, and let us see which cells form what structures in future. So the octane structure is arranged such that they form 2 layers or 2 tiers. So the upper tier cells give rise to the hypocotyle as well as the radicle, whereas the lower tier cells or the lower 4 cells give rise to the plumule and the cotyledons. So the upper tier cell, when they give rise to plumule and cotyledons, it depends if it is a monocot plant or a dicot plant. If it is a monocot, it will only give rise to one cotyledon, which in this case is known as a scutellum. Whereas if it is 2 cotyledons, it is the case in dicots. So now let's talk about the further divisions which occur, which give rise to specific structures. The upper tier cells gave rise to the plumule. So it is present at the tip of the cotyledon, and the next layer of cells are the ones which form the cotyledon. These cotyledons form the outer layer of the seeds, and then there is a hypocotyle region, then there is a radical region and a root cap region, and finally the suspensor cells, which help push the embryo into the endosperm. So finally, what happens is the hypocotyle and the cotyledons, they really increase in size, and as you can see, they are already forming a curvature. So as the cotyledons and hypocotyles increase in size, it can lead to further curving of the embryo, which gives it a typical horseshoe shape. It has been observed that embryogenesis in monocots is also in a similar pattern, except for the development of the cotyledon, in which case the monocotyledon is known as the scutellum, the terminal is known as the scutellum, whereas the plumule is known as the coleoptile, and the radicle is known as the coleorhiza. Now, in this video, we have learnt about the development of the embryo and the endosperm and how the pollen and pistil interact. The final product is the formation of a fruit and a seed inside the fruit. In the next video, we will learn how do the fruits and seeds develop.